We will get started. I guess I'll keep you. So I can't see you guys very well because my screen is a slideshow. So if people have questions, you can either put it into the chat, but I might not notice it. But so feel free just to turn on your mic and just say, hold on, Mark, I have a question. And I'll also have some questions I might throw out to you guys that just to have you guys chime in and see what you think.
Mark, can I ask you something?
Yes.
You are going to be able to let people in for this part, because some of my students told me they were going to come in, but I don't know if they are already in or if you're going to be able to let them in.
First of all, hi, Christina. Nice to hear from you.
Hi, Mark.
Yeah, so Luke has control and he'll let people in if people are showing up.
Yeah, there's no waiting room. So if they have the link, they should be able to enter.
Ah, perfect. Thank you.
Okay, thanks. Okay, so this workshop is about systematic reviews and meta-analyses, and I'll describe what these are. And so while this might seem like it doesn't pertain directly to what we've been talking about today with urban invasions and collecting data on non-native species, there is an important role for us to do some more synthetic work to look at studies that have already been published to try to answer some of these broad questions that might be of interest to us as this group or you as individuals. And so I know there's a good number of graduate students and postdocs also on this call, in this workshop. So this is hopefully you find this valuable for your forthcoming projects as well. And one thing I'll say is that, so this is something that I've produced, but I've also used material from someone named Jérôme Pellet, who works for a company called N&P Biology. And I'll just say as an aside that a group of us have created teaching materials associated with conservation and environmental management that are all going to be available soon on a resource called Applied Ecology Resources, and this is one of them that's on there. So reviews, meta-analyses, these synthetic efforts are extremely important. They're extremely important because they help define the state of understanding on a topic, right? As powerful as individual studies and experiments are, nothing is as definitive or as convincing as a synthetic piece that looks across a large number of individual studies. What a good review or meta-analysis should be is that there should be a timely overview of an area of research or a specific question, and they should also help guide future research. They can also be quite valuable for providing guidance for management and policy, but they're also really valuable for an individual's career. So especially for some of the early researchers in this workshop, it's a great way to get your name associated with a specific area for establishing your reputation as someone who works actively in, knows a lot about a specific topic. And also they're important for your career because they tend to be cited more than other types of papers. So really good reviews and meta-analyses, because they're seen as a definitive state of knowledge at a particular time on a topic, they tend to be quite cited by other studies that are exploring a similar topic. There's also a need, just a basic need for research syntheses, because think about it, there's in ecology alone, there's more than 12,000 articles published every year. And so if you were to read 2 articles per day, as would be really ambitious, you would fall behind by about 17 years in a single year, right? So there's no way you can keep up with all the literature that's being produced. And to try to keep up with that literature is just simply not feasible. And you can even think about just the top journals in our field or your specific field and ask yourself, have you read the latest issues of Nature and Science and PNAS and Biological Conservation, Conservation Biology, Journal of Applied Ecology, Ecological Applications, Trends in Ecology and Evolution, etc. So you can already see that just even in the top journals in our area, we can't possibly keep up and read all of them. So that's why these single papers that are really synthetic and help guide and understand single research topics are really valuable. So you notice that so what is the main difference between traditional review versus a research synthesis? So I've been using this word systematic review and meta-analysis, and I'm going to get a little more into defining exactly what those are. We have a long history of reviews being done in ecology, but we don't have a very long history of systematic reviews being done. We have a little bit of history of meta-analyses. So traditional reviews, as most of us have read, tend to be very broad in scope. They tend not to have overly specific questions. Their goal is to say, what is the state of our understanding about competition, for example? They tend not to be very clear or selective in the materials that get included. So traditional reviews will go through and talk about different experiments that might have manipulated something like competition and what they've found, but you don't quite know how unbiased their sampling was, whether they just looked at a few journals or what. They tend to be very qualitative, which is they'll describe the outcomes of some really influential studies. What is influential? I don't know, but it's what the authors have decided are influential studies. Whereas we're moving now into an era that we have so many studies available that really good practice and a commitment to open science requires us to do things in a little more of a systematic way. And so to do this, we often need to have very focused questions. We need to have very explicit and comprehensive strategies for searching literature and evaluating that literature. We need to have criteria that's uniformly applied across all sources of information. And there needs to be a quantitative element to it as well. So the research synthesis types we can think of as either being qualitative or quantitative. So qualitative would be the traditional narrative review that we're all used to. But that also includes systematic reviews. And I'm going to talk a lot about what a systematic review is, but they're meant to be very rigorous and very transparent in their methodology. And quantitative syntheses could include vote counting, and I'll describe what that is, but basically the number of studies that show a pattern versus not. And meta-analysis, which is a robust statistical type of analysis, but to do a meta-analysis properly, much of what I'm going to say about systematic reviews is required for meta-analysis, right? There needs to be rigor, there needs to be clarity, there needs to be repeatable methodology, there needs to be transparency. So I'm going to go through the 7 steps for conducting a review / meta-analysis. And so all that I'm talking about right now is really about a systematic review or a systematic meta-analysis. And you can see the 7 steps here, and we'll go through each one, go from the very first part, which is forming your question, all the way through to the end to how you report and disseminate that information. So question formation. There's things that you need to keep in mind when you're forming your question. And so just like if you're designing an experiment, you're going to, before you start that experiment, you're going to refine your question, have a clear question that's answerable with an experimental design, and it's clear what the control and what the manipulation should be given that experimental design. It's also clear what you should be measuring given that question. And the same needs to be true for systematic review and meta-analysis. So it must be an answerable question. It should be currently unanswered, or maybe that's a little strong, but at least at best it's partially answered or partially understood, but more information is needed. If it's a broad question that includes management elements, you should be thinking about designing these questions in collaboration with other people in or outside of academia. Questions should be unbiased or worded neutrally. So you shouldn't say, is this good for this? That's not an unbiased question. The unbiased question should be, what is the effect of this on this? because that can bias the perception and the outcome. And certainly if it's an applied question, could bias the perception and uptake by practitioner or other stakeholders. And also, a good question will really guide how you search for information. So here's a question. How can we reduce the impact of alien invasive species on native biodiversity? So let me ask you guys, do you see any problems with this question? Does anybody see anything that's not quite right?
It's too broad.
Yeah, so it's too broad. But this is not unreasonable for what a traditional review might be titled. But it is too broad. You can't really design an experimental design around it. So how would it be changed? Anyone have an idea what an easy change would be?
Divided in small pieces.
Sorry, can you say that again? You broke up for a sec.
We can make small pieces of these questions.
What do you mean by small pieces?
Maybe to think in the pieces that that the alien invasive species have as an impact. Then if we make some small pieces of this question, we can search the information we need.
Yeah, exactly. You can kind of subdivide it into different types of impacts or different species. So let's go up. So we already did that. So it was too broad, poorly defines species and elements of biodiversity. So those are the things that we can break apart into smaller pieces. And so let's go to this question now. So this question is poison baiting effective at eradicating rats from islands? So this is a much better question. And the reason why it's a much better question is that we have a population. So we know exactly what we're talking about. We're talking about rats from islands. We have an intervention, a specific intervention, which is poison baiting. And we have an outcome, eradicating. So this view of a question, or as a title of a paper, is called the PICO formula. The PICO formula is designed to build effective questions. And PICO stands for population, intervention, comparison, and outcome. So in that title I showed you, right, the population are rats on islands. The intervention is the poison baiting. The outcome is the eradication. In these questions and titles, we don't necessarily see an explicit comparison or control, but it should be implicit. It should be obvious. So in that title, the obvious comparison or control is no baiting. So that has to be part of the question and the methodology. So once we have a question, the next step is that we need to design a protocol. So it needs to be a protocol formation. And so this needs to be developed before you start the research. It acts as a guide for conducting the systematic review or meta-analysis. It clearly lays out the methodology and is finalized based on agreement between researchers and other stakeholders who are involved about what the methodology should be. And it should be openly shared and made available, which I'll get to in a little bit. So the key components include, right, the formal presentation of the research question, so that should be clear. Generating some background information, being clear about the search strategy and criteria for screening articles, methods that will be used for appraisal and data extraction and synthesis. So a lot of this stuff is actually available on a website called the Collaboration for Environmental Evidence that gives good guidance on designing these protocols. As I said, I'll come back to this about what you should actually do in a bit at the end of this talk. Sorry. All right, so once we have a question, we're designing our protocol. The parts of this protocol then are making decisions about the various steps. So searching for studies. So, where should we look for studies and information? Well, the most important thing is always to use a diversity of sources for your search. Don't just rely on ISI or Google Scholar or something like that, but try to use a diversity of information sources. So online literature databases is the obvious one that we all use. Trying to find great literature, which traditionally is quite hard to find, but in 2 weeks it'll be much easier. I'm one of the designers of something called Applied Ecology Resources, which is going to be a gray literature repository to make gray literature searchable and findable. So that part will become much easier, but just think of it this way. When somebody in the medical sciences does a systematic review, they are often getting case studies and reports from gray literature, and those often form important sources of information, as they should in ecology, especially for applied type questions. You can also do more general web searches. You can also look to individuals or individual papers and look through the bibliographies. You can look at papers that are citing key papers, for example. These are all legitimate ways to find information. You just need to be clear about the methodology beforehand. And it's important to look in more than one place because any of these places could be biased. So for example, if you're just looking at ISI listed journals, there might be a bias towards positive results, so papers that show significant results, and those that may not have found strong results could be buried in other places. So the databases that you search can vary depending on their discipline and the types of material they present. They can vary in whether they include books or proceedings from conferences, where they include dissertations, right? So we often always just go straight for databases that include peer-reviewed journal articles, but there are other legitimate sources of information that may not be captured by those, and it's important to keep that in mind. It's also important to keep in mind that even if you think you're doing a fully unbiased search, there's still going to be biases that you cannot overcome, such as language biases. And so you're going to be looking invariably at articles in English and perhaps one other language. But there might be a lot of material out there that you're simply not capturing. And just in some ways, sometimes there's just no way over that, but you need to be at least cognizant of it. So when searching for literature, you should document everything you're doing. You need to be transparent. And the idea is that someone else should be able to come along and use all of your steps in your methodology and get the exact same or very close to the same information that you've received. And so there should be a record for each source. So you should have a full list of the search terms that you used and how they were combined. There needs to be the name of the resources and databases that you used and the dates that they're searched, so that somebody can go back and set the database to that date and redo your search if they wanted to. If you've changed default search settings in that database, so a lot of people go in and change some default settings in Google Scholar, for example, just need to state whether or not you do that. Whether you're searching keywords, topics, titles, abstracts. So you need to be clear about that. How and when you remove duplicates, and a list of the number of and retain records of all the results that were returned by searches. And so you need to be cognizant, like I already said, of some of these gaps that are going to appear. And if you've decided to try to address some of these gaps, again, you need to state that in the methodology. So for example, for journals not covered by ISI, you made an explicit decision to search some other resources. You need to be clear about that. So your literature search, if you go back to the PICO formula, so the population, intervention, the outcome, those aspects should appear in your search terms, right? So you want to have that rat example. So you want to have rat somewhere in there. You want to have islands. You want to have eradication. You want to have poison baiting. All those terms should appear in some combination of search terms. They don't have to appear all in the same search, but all those different aspects are important to your question and answering your question. So they should appear in your search strategy. You also need to consider synonyms and alternative spellings and abbreviations, especially in this the topic of this meeting, which is non-native species. Some of you prefer aliens, some people prefer exotic, some people prefer non-indigenous, some people prefer invasive, right? And so that's examples of synonyms that we all need to be aware of. You need to, be aware of some of the trade-offs that are involved between how much effort you put in your return, right? Those first few searches are going to return a lot of stuff, but every refinement after that is going to return fewer and fewer new items. So you need to make sure you know when to have a cutoff. And you need to have there's a trade-off between sensitivity and specificity. So if I just put in a general word like invasive species islands, obviously I'm going to get thousands of articles. So that's not very specific, and I'm getting lots of stuff I have to wade through. I could be too specific, right? I could have a search term that's the Norwegian rat, Hawaii poison, and I might only get back 2 papers, right? So there needs to be some appreciation and some fine tuning of the search term. And this, you can do this beforehand. You know broadly what you want to do. You can play around different search words and decide, some of these are too broad, some of these are too specific, but there's kind of a sweet spot here. And that's where I'm going to really explore and develop my methodology around. The next step is for screening articles. And citation managers can help with this because they can easily identify duplicates. But you need to be clear about how you're identifying and removing duplicates. You also should, even when you're excluding studies, you should keep a record of those that you've excluded because there might be some interesting information in those that someone else might want to see. And you need to have a set of what we call filters. So how you're going to filter. You're not going to filter by reading all the papers that were returned in a search, right? So if you do a search and you get back 1,300 articles, you're not going to read every single article. So the first level of screening should be the title, because the title should be informative, and there's going to be papers that are included that, at best are marginally related to what your question is, and you can often really identify those right away. So for example, if you have search questions about rats and islands poison baiting, you might get back articles about mongooses or snakes that are eating rats or something like that. And the title might tell you that right away, so you can exclude those. Next would be to look through the abstracts to make sure that especially if the title is inconclusive or sounds appropriate, you look through the abstract and you might get confirmation that it is appropriate or that it's not appropriate, especially if you've decided to not include other reviews, right? So in a meta-analysis, you probably wouldn't include reviews. And so the title might not tell you if it's a review, but the abstract certainly should. It is good practice to have more than one person look through some of these articles on a random subsample. And we'll get into that in just one second. So when we're filtering, what are we considering? We're considering whether the subject is correct, the intervention is correct, the measure and the outcome are correct. Well, I guess correct's not the right word, but is appropriate to answer your question. And so as I said, this should be validated by 2 people, and you can assess this validation using what's called a kappa statistic. And so what you're basically doing is you could take some random subset of articles that were captured in your search. You get 2 people who both know the criteria, this question, to go through and include or exclude them. And then you compare the 2 in something that looks like this table right here. This is Cohen's kappa statistic. And so you have reviewer A, reviewer B, and you ask how many times were they in agreement? How many times were they not in agreement? And you can come up with a statistic. And the kappa statistic, you can see in the bottom, between 0.2, sorry, my dogs are barking. Between 0.2 and 0.4 is fair, 0.4 to 0.6 is moderate, and above 0.6 is considered to be a really good agreement. So if you have a second person go through a subset of articles and there's high agreement, then you can feel confident that the way you're approaching them is valid. It doesn't mean the other person has to be a full member and is going through all the articles as well. It's just a confirmation. So you could, so let's say you get a return of 1,000 articles, just randomly select, 24 or 30, have yourself, another person go through them with a clear description of the criteria. If the kappa statistic is high, then you feel confident that you're on the right track and that you can move forward. All right, so how we evaluate articles then? We use something called the CRAP test, which is ingeniously named test. The CRAP test stands for currency, relevance, authority, accuracy, and purpose. And it's basically asking yourself these different questions. So what is the value of something? Well, is it current? So, is it something that is timely, has been produced recently? Is it relevant to your needs? The authority, this is really important for gray literature. Who's producing the information? The accuracy, well, actually, let me go back to authority. Nowadays we do have a bit of a crisis in peer review literature because of all the predatory open access journals that exist that are willing to publish things with minimal peer review. And so if you're searching, especially outside of ISI, and you get back all these articles from these journals, it is good to go and look at the journal and make sure that they are in fact a valid journal and they're not listed on some predatory journal list, or they appear to have an international expert editorial board and they seem to publish articles, meaningful articles on that topic. You also want to assess the accuracy, so the reliability, truthfulness, correctness of the information. So if it seems like something's wonky in the study or they have a sample size of 1, maybe you want to score a low accuracy. And you also want to assess the purpose. So I'm just going to click on this link here to open the CRAP test. And you can see there's actually worksheets for this test. And you can see it's quite long, right? And so I wouldn't suggest that you necessarily go through this on every single item. If your review returns to you 45 articles, maybe you can go through this quickly for each article. If you have hundreds, you don't want to. But what I would suggest is the first few you go through using this, and then in your mind, you start immediately scoring papers given this information. So you can look at it quickly and say, it is relevant. I would score it high on this. The authority, yes, it's a good journal. It's it's scientists that produce this. I give it a high score. Accuracy. You just start thinking this way, which can really help you. And so in your methodology, you can either say we use this score sheet to assess every article, or you could just say we use the framework of the score sheet to assess articles, which means you don't necessarily have to go through and look at them all and score them all in detail like this. But it is a good way to set your frame of reference. There's a number of existing tools to help you assess papers. So here's another one. It's called the Prisma checklist. And this is built for transparent reporting. So if we click on that one, you can see it's a similar type of set of information. It's not really for determining the validity of a literature item that you're going to use in your systematic review or meta-analysis. Instead, what it's about is really about where the information lies in the article and whether it supplies that type of information to you. And so you can see as you scroll down, again, it's kind of long, but it tells you that whether or not the individual article is conveying to you the rationale for this study, if how they're collecting information, how they're reporting information, how they've selected studies. So you can go through this and assess. Again, if you have hundreds of articles, you wouldn't want to go through this with all of them, but by going through and understanding it, it becomes more obvious the types of information that you want to review for yourself. But I guess the point is just to know that there are these different toolkits out there to help you establish transparent criteria for assessing articles and extracting information from them. So coming to data extraction. So data extraction is something that is best done when you're appraising the study quality. So you're starting to go through the study in detail for the first time. You're trying to establish the quality of the study, how valid it is. You can put you can put a confidence score in that study, but at the same time, you should be extracting information from it. And the best way to do that is to construct a spreadsheet for yourself. And I'll show you an example of what that looks like in a second. But you need to be aware of different elements of the study and the information they're presenting. And you want to make sure that you're aware of things like the location of data in the article, how you're extracting information. Is it coming from tables? Is it coming from figures? You need to note if you're doing calculations or transformations yourself, or if the study did them and you need to do calculations or transformations to get things standardized. You need to know, keep track of whether you're getting information from appendices or other sources, whether you had to contact the author directly to get information. So in your database, each row should be a single study or experiment. And I say that because some papers report more than one experiment. So example, a paper might run an experiment in a deciduous forest and run the same experiment in say a pine forest. And you can treat those as two separate experiments. They're analyzed separately in the paper and they appear as separate entities. It's one study, but it's two different experiments. That'd be two different lines, rows in your database. You need to include as many columns as possible. And the number of columns will grow as you read more papers, because you'll get to your fifth paper and go, oh, I should also include this because I noticed the last couple of papers have commented on the time of day or something like that. And so you need to have columns in your database for the design and methodology. They are necessary because you're going to come back and sometimes need to distinguish studies based on some fundamental methodological decisions that they've made. So for example, study location needs to be included, year, plot size, length of time that the study was run, number of species included, habitat type, etc. All those are important methodological considerations that could make studies not so comparable, or at least offer ways to group studies together. So as an example here, I have this one systematic review on the side here that's looking at boreal forest conservation of species richness in production forests. And they did a systematic review. So they went out and looked at this topic. And you can see they produced a figure that is according to different taxa. And you can see there's quite a variation in the number of studies they had by taxa. So they had lots of studies that looked at fungi and vascular plants, but not nearly as many that looked at, for example, lichens. And so these are important criteria that should put qualifiers on the generality of your answer. So you might find some general answers to the question they're proposing in the systematic review, but then you might say, but that was really informed by or biased by the number of fungi and vascular plant studies that were included. For systematic review, you need to indicate the directions of findings. Are they increasing? Are they decreasing? Is there significance? And it's also good in these databases to have a notes section. So you can insert some qualitative perspective. So you can have all these columns that have all these, simple entries of yes, no, 1, 0s, values, whatever. And then a notes column where you're saying, okay, they show this pattern, but they might have not indicated this, or it's, the figures they showed seem to have some outliers that might have been disproportionately influencing the results. Those types of notes you want to have in there. So here's just an example of just one of mine own systematic review meta-analysis projects. And you can see that, wait, let me get my pointer here. And so we can see here, for example, rows 9 and 10 are coming from the same study. And you can see one was in forest and one was in savanna. So they're listed as separate rows. I don't want you to pay too much attention to the columns, but you can see some of them would be common no matter what type of synthesis or meta-analysis you're doing, right? We have taxon, we have whether things are in different habitats, the country they're located in, number of plots, plot size, number of taxa. In this case, it was traits, so it was phylogenies and traits, etc. And the reason why we want to include a bunch of those different variables, if you look at this example meta-analysis here, is then you can start to split apart. So for example, same data, looking at all taxa, but then splitting it by invertebrate, plant, or vertebrate, looking at community level, population level. So you can start to pull apart the information and group it in different ways to say, okay, we notice there's really a positive effect here of, this is on introduced species across urban gradient, but it seems to be really driven by invertebrates, right? So that positive effect seems to be driven by invertebrates, not so much by vertebrates. So having those different categories can be really important. So for a meta-analysis, you're basically carrying out the same steps as you are for a systematic review, but you need quantitative values as well. So there absolutely has to be sample size. There has to be mean values from treatment and control or some other type of comparison. There needs to be measures of standard deviation or other measures of variation. If the design isn't one that has a treatment and control, then you might be recording regression coefficients, correlation coefficients, but there has to be some quantitative measure that allows you to determine the effect of one variable on another. The problem with doing meta-analyses is that the information is not made always so obvious. Sometimes you have to look in appendices. Sometimes you have to go to online data repositories. So they might have uploaded their data to Dryad, for example. You might have to contact authors, or you get the data from their figures. And so here's an example. This is called Data Thief. This is a program. It's a free program. And what you simply do is you take a screenshot of a figure from a publication. You bring it into Data Thief. And what you can do in Data Thief is you can specify where the 0 is, where the 100 is on the axis. You can specify the values on the bottom axis. And then wherever you point to on this figure, it will tell you that value relative to the X and Y axes. And so you can kind of steal the data in that way. So for example, if this figure here, if this public paper here didn't give me the correlation coefficient or the regression coefficient, I can still get the slope of this line through Data Thief, right? So if I know the bottom and side axes and I click a point here and I click a point up here, I can estimate the slope of that line. So there are ways to get information. Same thing if they're showing you, a bar chart, but they have bar with error bars on it, and they don't tell you what the means were or the standard error or standard deviation. In Data Thief, you can then pinpoint what the mean value is and what the upper and lower bounds of the error bars are. So if they don't give you that information, there are ways to determine it. All right, we have a question in the chat. Is that a loud, nervous laugh? Yes, absolutely. There's no, it's not like you're republishing their figure, so there's no copyright issue. And it's no different than if you took a ruler, you printed off the paper and took a ruler and really carefully marked where different things were. It's this more precise way of doing that. And so in your methodology then, so in my papers where I've done reviews and meta-analyses, I say I've used data thief to extract information from figures when the values were not presented in the text. And I usually say that with a disapproving look at the authors of the original study for not supplying the original values in the text. All right, so now we've collected our evidence, right? So we've collected either with a systematic review in mind or a meta-analysis or both, and I'll say something about that later. So you've collected all this information. Now, how do you synthesize it? So in we got another question. Mine looks like a best fit on scatter plots. Yeah, so that actually brings up a good point. So when you see these figures, you have to be, in you're pulling information out of these figures, you have to know exactly how they plotted those figures. So was it a regression line? Did they use a nonlinear regression? Did they, and if it's error bars on a bar chart, are those error bars standard error? Are they standard deviation? Are they some other measure of variance? You need to know those details when you're extracting information. All right, so evidence synthesis. Again, we talked about the narrative synthesis. This is really telling the story of the articles that you've collected. You can talk a bit about the characteristics of the study, the data quality. You can use tables to help tell that story. We have another question. So Ben Beiser just posted how to do this in R. I suspect he means how to extract information from figures. Is that right, Ben? Yes. All right. Yeah, so the narrative synthesis, even though we're saying it's not quite at a rigorous enough level to adequately test questions, should not be ignored. You can still tell the narrative part in other types of more robust types of syntheses. So you still want to tell the story that, a lot of studies are showing these patterns, but there are these things that we should be aware of to qualify them. In reviewing the literature, it's noted that there may be these biases in these results, that there seems to be, over distribution of studies in north temperate regions. And, so you still need to tell the qualitative part and to help, and that's really important for interpreting and informing. The quantitative synthesis is usually more desirable, especially in cases where we want to measure the effect of an intervention or exposure or a treatment. And so there's a number of different statistical models that are available. We can still use vote counting in some ways to paint the overall picture of the evidence synthesis. So, if 90% of studies are showing a positive significant effect, that's a really important thing to communicate. If half are showing one thing and the other half are showing a different thing, that's also a really important thing to communicate before you get into analyzing any information. So vote counting, just to be clear, is just basically counting the number of studies that are reporting significant results, or if they're reporting a positive result, negative result, whatever, but just counting them. But it's important to note it has low power, right? Because it's not reporting sample sizes, not a mean effect size or anything like that. But it does supply some information. And as I said, especially in the cases when we have clear splits, it's really interesting to know that half showed one way, half showed another way. That could tell you something. So in the core of the systematic review, it's good to use tables. And so I've just shown this table from the Millennium Ecosystem Assessment. I think this is a really good table. So this is not quantitative, right? But it's conveying a lot of information. And so this is, in this table right here, it's talking about how drivers might be affecting different biomes or habitat types. And it's talking about the both the direction and the magnitude of that impact using colors and arrows. So there's actually a lot of information right here. We also see with Millennium Ecosystem Assessment or IPBES reports, we'll often see the degree of confidence in certain results. So they might show arrows of impact, you know, it's increasing over time, not increasing, but they also might indicate the degree of confidence in that result because of the available evidence. So some of those might be, we're really confident because of lots of evidence that that's happening. Other ones, we might not have that much confidence. And so in a systematic review, you really want to be saying, you know, in what cases are we finding, for example, these positive effects, these negative effects? Where are we finding cases where we're seeing highly significant results and not? What is the strength of our confidence, and how is this maybe influenced by sample size, number of studies, plot size? So, you know, for example, if you're doing a systematic review and you're dividing your studies into those that were done in meadows and those are done in wetlands, and you realize the meadows studies, there's just many more studies and they have larger plots and done for a longer period of time, you might communicate that you're more confident in those findings than in wetlands that have been done with, say, small plots and over shorter periods of time. So you might identify that more work is required. This is an example from one of mine recent ones where, let's not worry about exactly what I'm doing in these, but you can see here what I've done is, this is basically a vote counting over here. I've talked about the cases where there's these positive correlations, non-significant, negative, and mixed, which was I won't go into detail here. And then using the same color coding, I actually show what the distribution of coefficients were. So same thing over here. I'm talking about phylogenetic signals, the number of studies that failed to find them in red and did find them in blue. And then I'm showing the distribution of different measures of phylogenetic signal. And again, color coding by those values, just to provide more information that can help in the assessment of the question that was trying to be answered. And so I'm showing this just to show you that, so back here with the table, and here are some figures, the use of color and different types of information, so vote counting and actual correlations, taken all together, you can actually present a lot of meaningful information in relatively few figures that then take a lot of text to digest and talk about. So, so in those cases, those were essentially the core of how you might present the findings of a systematic review or meta-analysis, or systematic review. When doing a meta-analysis, you need to go a little bit further. You need to go beyond the vote counting, beyond the yes or no answer. You need to really be analyzing the effect of a treatment on mean values, so mean population size, mean species richness, etc. It needs to take into account sample sizes, and it can provide a very important basis for evidence for making a conservation and applied decisions. So to do a meta-analysis, you need to decide on the correct type of analysis. And it's really based on what the quote unquote common currency is. So what is the common type of data result that's being reported in these studies? So if you're comparing two groups, like a treatment and a control, or results from a large islands versus small islands, or pre-burn versus post-burn, or something like that, then you're comparing groups. And if the dependent variable is a continuous variable, like species richness, abundance, biomass, then you're really looking at Hedges D, or the response ratio. I'm not going to get into too much detail about what these are, but just so you know that there's these names out there and they're easy to find. If you're comparing two groups in terms of categorical response, right? Like yes or no. Then you use odds ratio. And if you're looking at relationship between two continuous variables, then you're really just looking at correlation coefficients. So Hedges D, I'm just showing it here because just demystifying it. So what you see here is the equation right here. It's a really simple equation. And it's basically the average effect of an experimental treatment. When I say experimental treatment, it doesn't necessarily have to be an experiment where somebody went out and added nutrients or removed something. It could be, like I said, post-burn, pre-burn. It could be an invaded site, a non-invaded site. So they weren't manipulated in an experiment, but they are something that you're contrasting. And then compared to a control. So for example, we could have two green spaces. One has been invaded, one's uninvaded. That would be an experimental treatment in the control. So you have mean values of those, and they're standardized by the standard deviation. And sometimes there's an adjustment for sample size that could be really meaningful. And then from Hedges D, you end up with these values that tell you the magnitude of the effect. Response ratio is very similar. You have the mean effect of an experiment, mean effect of a control, and you're just taking the natural log of the experiment divided by the control. So it gives you this ratio of response. Odds ratio, as I said, it's categorical responses. So you have a treatment and control. You have response, no response. You have a yes, no. Yeah, nothing more, nothing quantitative more than that. But you can do a very simple calculation to come up with the odds response ratio. That tells you the odds of some response in a treatment relative to the control. So yes, no could be, is the species present or absent? We have a question. Question for later. All right, I'll put a pin in that and we'll come back to it later. There are a number of really useful materials online for doing meta-analyses. So there's useful packages like Meta4 are two commonly used ones. There's also wonderful books and guides now available. And I've pointed to one here. It's called Doing Meta-Analyses in R. It's actually a really fantastic resource. So if I just click on it real quick, I'm not going to go through it, obviously. But you can see on the side here, there's 17 chapters. And if you click on any one of them and you click through, you can start to see that it gives you the R code and the example. So you can actually go through the R code and reproduce what they're doing. And so you can design your own analysis after that. But it's a really great resource and it's free. So it's definitely something you should look into. At the end of the day, what you often want to see with a meta-analysis are results that look like this. And those R packages will produce these types of figures. Not as fancy as the one I've done over here on the side with these little graphics in there. I prettied that up myself, but the basic result of these mean effect sizes and estimates of standard deviation, and then the overall effect size, you can see in both these cases. This one's an odds ratio and this one's Hedges D, and you can see they both look quite similar. They're producing information similar. So what you typically see is a vertical line, Hedges D at 0, odds ratio at 1, which basically tells you if there's no effect in whatever comparison that it is. And so, for example, if we look up here, we see rural versus urban in the population size of exotic species. We can see that there's a positive effect. The arrow bar doesn't overlap with 0. So that would tell us there's an effect. Whereas natural versus rural, there doesn't seem to be a big effect. So this is a nice way to show you. Here, I'm assuming that these symbol sizes on the odds ratio one are scaled to sample size of those individual studies, which also helps give you some more information. You can group these studies on the side here, like I did, by grouping by whether they're natural versus rural. It could be by geographic region. You could group them like North Temperate versus Tropic. You can group them by like, savanna versus forest. You can group them in ways that make them quite meaningful. You can color code them. There's lots of different things you can do in this figure to reveal a lot of different types of meaningful information. It's also really important for both systematic reviews and meta-analyses to be really clear about forms of bias that can creep in. And there's always bias. And you know, we think of bias as a negative thing, and it is, but it's not necessarily a damning thing. It's not going to result in your paper not being published, because there are biases everywhere, and it's just important to acknowledge them and to make suggestions for future studies how they could overcome those. Some of these biases can be corrected for in analyses, but sometimes they can't be, and they just need to be commented on. So biases, for example, there are publication citation biases, where studies with low significance might not appear in top journals that are easy to find. So you may be biasing by finding stronger studies than if you just do random studies around the world. There's language biases, as we said. There's familiarity bias. So you're more likely to be selective of articles of topics that you're more familiar with. So that can bias. And here I'm showing an example of a geographical bias. So for example, you know, this is not an uncommon pattern to see when you're collecting the results of studies, that you notice that they're all coming from North America, Western Europe, and Australia, for example. And that's something that is, you know, increasingly less so, because we're getting, you know, more globally distributed studies appearing, but it's something that you need to be cognizant of. And if there's no correction for it, then you need to be able to comment on it, and comment on the need for more studies in other parts of the world. All right, so when it comes to reporting and disseminating, so the elements that you need to include in your report, and how you're interpreting, how you're evaluating, and how you're concluding from these findings, is that you need to talk about the reliability and the quality of the studies. You know, are you dealing with a lot of studies with low replication, small plot size, really short duration? You can say that. You can say we need more longer term studies. The relevance of the study, are a lot of studies like kind of close but not precise? Do we need more research in the specific topic areas? How consistent are those findings? You know, even though the overall effect might be a positive one, there's actually a lot of variation. Why is there that variation? So you need to talk about the consistency of effect, what might be influencing it. Yeah, you need to also consider other plausible explanations that could also be explaining the patterns that you're observing. And finally, when you're writing a review, so unlike research articles, so primary research articles where you're doing an experiment, the format of review tends to be a little more flexible. So some are written like a research paper with a, you know, introduction, methods, results, discussion. And that's typically the case with meta-analyses. But quite often they're also written more like an essay format, where they're a little bit more informal in their structure. But I would say that the more recent move towards more transparency with systematic reviews means that there should be more concrete structure to the articles. So there should be a section on methodology, even if it's not a meta-analysis, your search terms, etc. All that needs to be reported. There should be an introduction with strong hypotheses. So the more that you move these reviews towards looking like a traditional research paper, the better it is for communicating information and making it more transparent. When they're just essay format, they're hard to reproduce. Whereas when they have clear methodology section, they're much easier to reproduce. I mentioned this earlier about these guidelines that exist. And so this is from the Collaboration for Environmental Evidence. They keep updating their guidelines. They have very, very precise and clear guidelines about doing this type of synthesis work that lays out a lot of things that I've talked about in this workshop about how to go through these different steps and these criteria. We have another question as well. You say something about sample size. I'll come back to that one in one sec. I'm just about done this part of it. So I'm coming back to one topic that I kind of hinted at earlier, which is what to do with your protocol. So we're having this move in science towards greater transparency, and it's becoming good practice for both transparency and repeatable science, which means making the methodology and the research plan open and available. So we've seen the recent emergence of what's called research registration or protocol registration as a paper type, and this has clear benefits. So in the journal Environmental Evidence, they have what's called a systematic review protocol, which is basically, well, I say it a little bit later on, but basically it is the introduction and the methods section of the paper. It goes through the generation of the questions, the background information, outlines the methodology. So all the details that we outlined in this workshop, such as search terms, selection criteria, all that kind of stuff, lays that all out and publishes that. My journal, Ecological Solutions Evidence, we also have registered reports. And so stage 1 is the same thing. And it could be for a review or an experimental design, but it's again, it's basically publishing the introduction and the methods. It counts as a peer-reviewed publication. It appears as a searchable peer-reviewed item. In many journals now, and this is driven a lot by the medical sciences, but we're now seeing in ecology. So in Ecological Solutions and Evidence, we have a stage 1 and a stage 2. Stage 1 is that pre-registration. So it's the introduction to methods before you carry out the research gets published. The journal is then committed to publishing the full article, which is called stage 2, regardless of the strength of the outcome. So if there's no interesting findings, then it still gets published. And so you get a second article as a follow-up, and it shows that you're really committed to open, transparent science. All right, so that is the end of the presentation as I have it. So I'm going to stop sharing and I'm going to start answering some of the questions that we have that appeared in the chat. So the first question was from Tiziana. Which approach is best when the studies are using different categorization levels, spatial scales, and they're using different ones? So that's a great question. When all the studies are using, let's say, different plot sizes or slightly different ways of categorizing things. So let's say they're measuring disturbances, they might have different thresholds for what a disturbance is, or they might have different definitions of land use type in an urban setting. So at the end of the day, you need to make some decisions about the grouping variables. And the decisions need to be informed and need to appear as though that they're objective. So what I mean by that is, let's say they're all using different plot sizes. You could then say, well, I'm going to have a grouping of small, medium, and large plots. And my thresholds for defining those are these thresholds based on what I know about these systems or about how number of individuals are organized at different spatial scales. So you can group them in that way. Land uses, again, you could group them. You can say, you know what, there's a bunch of these different land use definitions. I'm grouping all of these into highly anthropogenic. I'm grouping these into moderate. I'm grouping these into low anthropogenic or more natural. So those can be complications, but there might be ways that you can kind of supersede them and group them together based on some meaningful information that you have. The second question, can you say something about the sample size of the review of the meta-analyses? How many independent studies is too low for a meta-analysis? How many would be valuable or publishable? All right, this is a great question. And actually, this brings up a point I wanted to make. And I'm actually going to share my screen again, because I just want to go back to one particular graph that I showed from my own meta-analysis. So this one right here. So this one I published. These ends are sample sizes, and some of them are horribly small. So I'll talk a little bit about that. So just remember, there's some of them are like sample size of 2. so what we did in that case was we originally designed our study to be a meta-analysis. And we thought, oh, there's going to be a bunch of studies that measure the population size of non-native species across very particular habitat types across an urban gradient. Well, what we found was when we started doing it was a lot of studies were not even stating what the plot size was. They were just saying, these are the number of non-native species we see in parks. This is the number that we see in yards. And we couldn't use those because we didn't have enough information that allow us to compare them to other studies. And so we had to exclude them. And so ended up with some really small sample sizes. So what we did in that case was we actually did a hybrid systematic review meta-analysis. And I've done that at least twice now where that ended up being the outcome. And it ended up being the outcome because you do have lots of evidence, right? There are a lot of studies that are evaluating, in this case, the average population size or the number of species that are in parks versus yards versus woodlots. And you can tell that story through a systematic review. A bunch of studies found that there were more species and or higher abundance or more species in these contexts, but they weren't rigorous enough to do a meta-analysis on. We couldn't combine them in meaningful ways. And so we told the full story. We did the review, and then we showed some of the meta-analysis. Then a big part of the end section was why we couldn't include a bunch of studies in the meta-analysis, and it was a call for doing better, more precise science on some of these questions. And so one of the things we came forward with was a recommendation that when you are doing work on non-native species in urban contexts, you need to have fixed plots of a fixed size. You need to quantify what type of habitat they're in. So for example, it's not just enough to say, and a lot of them would just say urban and less urban. We don't know what that means, and we can't compare that between two cities. But if you say these plots of this size were in an area where there's this much impervious surface or this much housing density within a 1 kilometer radius, then we can work with that and we can compare to other studies. And so a big part of that study then was, yeah, we have lots of evidence that we can say something about it, but we need more rigorous data. And here are our recommendations. Future studies going forward need to do this and this and this. And that was actually one of the origins for GUBIC, was that actual exercise of a systematic review meta-analysis. We realized that we were just lacking some really meaningful information that could be compared across different studies. Are there other questions? All right. Menelik says, does an automated method of literature review fit into all this? Maybe acting to assist in the literature filtering. This is, yeah. So automating some of this. That's an interesting point, and I kind of feel like we're not quite there yet in the sense of the tools are emerging, so you can use some sort of AI learning algorithm to do some of this for you, but I feel like that would be a project unto itself. It'd be a separate project to get it to work. But you could use some automated processes to filter out, but you'd have to do some quality checks to ensure that they're working well. And then Francois posted something that is probably assessing this.
Yeah, it was just something that popped up today, this week maybe, of sort of a new way to assess evidence, since these, well, there's been some discussion, I think, lately about the types of sample size that we might wish for in ecology for meta-analyses, but that even small meta-analyses have a lot of impact for practice. And so that even when it might not be enough to sort of make a clear conclusion on a scientific question, it might still be enough to improve local decision making.
Yeah, that's a really good point. I mean, I guess in that vein, I mean, one single well-designed experiment or study could also inform policy and management, certainly. And so a small meta-analysis should be even more valuable than a single study. So there should be a real value. Oh, I think Basil wants to add something also. Basil, do you want to say something? Wait, you're muted.
I was just wanted to say that, you know, regarding the automation, you already answered it, that, you know, I've done some research. You can answer like questions about like how many studies have looked at concepts using automated content analyses, but you can't really get at like quantitative effect sizes with that. And you have to do a lot of validation as to whether or not your concepts are being identified correctly. Yeah. So
Yeah.
I'm happy to discuss that. I have a colleague of mine who's really good at that, Gabriela Nunez Mir too. So yeah, if anyone's interested in that.
Yeah, I guess if you worked in an area or question type where everyone presented the information the exact same way, then it's much more feasible and realistic. But in reality, in ecology, you know, one person's going to, you know, bless their heart.
We've had good success, like looking at thousands of literature papers for like forestry research, and you can come up with like exploratory analyses of what concepts have been looked at over time. But, you know, these sort of effect size questions are not achievable with that sort of approach.
Yeah, and so some studies will present like an ordination, another one will present, you know, bar charts, another one will present mean effect sizes, and another one will do regression.
Yeah, you can't do that, but you can identify what concepts are being discussed. You know, that's about it, and the frequency of that. So
yeah. So I didn't talk about, I think I had one slide that mentioned it, but there are ways of doing text analysis to look at concepts and terms, but even that can get really complicated because you need ontologies. So you can have, for example, you can have the word biodiversity and ecosystem function in a sentence, which means something different than biodiversity effects on ecosystem function, right? And so even text analysis can get really tricky fast, but there are ways to pull out how often concepts are and if they're related to one another, like X affects Y, or there's a positive effect or something like that. But, you know, if you're dealing with a relatively small number of papers, like a couple hundred or less, probably not worth the investment to figure out.
yeah,
That's
train, correct. Yeah, you're right.
To train these methods to work.
Yeah.
Ben?
Yeah, I just wanted to speak to the small sample size. So in my experience, as others have said, you know, you can get a lot out of you know, a relatively small sample size, especially when it's in your field and it's a big question and everyone keeps citing the same, even if it's a small number of papers, 7 or 8 or 12 papers, you know, oh, this has been looked at, some authors have found this, and you're like, wow, I would, you know, is there an overall answer too? Like, you know, I did this with, you know, is there a difference between native and invasive species area relationships? Because everyone always cites, you know, these 5 papers found this, these 7 papers. And so I just asked myself, like, we don't have a simple answer. There's been a decent number of papers. Like, can we just pull it together so we could say, yes, we found generally there is, instead of having to say these 5 found a positive, these 5 found no relationship every single time. Then you kind of know in yourself, just, wow, it'd be great if we had something that could just summarize this, give us the current state of knowledge, show where we're missing some things. So then we can kind of focus on that, building that out, instead of, like I said, having to just run through a ring of all these papers. And, you know, and so that's kind of what led me to kind of say, okay, it's fine. You know, we might not have a huge, I mean, if there's 3 papers, you don't need to, but 10, 15, 20, even though it seems like a relatively small sample size, synthesizing that at least can be really useful to help, you know, get the big picture and kind of move on. And so that's what I've had in my experience.
Yeah, I agree. And I think you also highlight something else that is a potential concern, is that when we cite papers as evidence, I mean, I do this, everyone does this. You'll cite a paper that the other paper cited as evidence for that thing. And you know, it is sort of this founder effect of citation where certain studies just start getting cited as evidence for this thing, and they keep getting cited as evidence for that thing. But in reality, there might be 15 other papers out there that have assessed that same thing, but may not totally agree with that one paper. And so even with small sample size, Ben's right, just by going out and doing the act of getting all these studies, comparing them and assessing them, can you come up with a more valid assessment of the state of knowledge on something. So people are doing a really good job of listing different types of tools for determining citations and all that in the chat. I have to remember to save it. Luke, remember to save this chat when we close down. And then Dudu said Atlas TI. I don't know Atlas TI. Does anyone know Atlas TI? I'm looking it up right now. Quantitative data analysis research software. I've never used it. It looks like a proprietary one you have to pay for. Oh yeah. So just from their screenshots, it looks like they do citation networks and stuff like that. So yeah, I don't know that one. Commonly used in sociology. I see. Yeah, so there are probably lots of tools out there. I am always biased towards just looking at things in R that are available in R. So there's probably other options as well. Any other questions, thoughts, experiences? Cool.